In the previous video, I spoke about transcription initiation in a bacterial system. Here, I am going to talk about the stages of elongation and termination. Let us recall the penultimate slide of the last video. We have seen that in spite of the occurrence of abortive initiation, once the RNA polymerase is able to synthesize about 15 nucleotide long RNA, the sigma factor is released. The polymerase escapes from the promoter and proceeds towards elongation. The elongation complex at any instant of time consists of an 8 to 9 base pair RNA-DNA hybrid within an 11 to 12 base pair of melted DNA bubble. On the upstream side of the hybrid is single-stranded RNA moving through the exit channel, and on the downstream side of the bubble is double-stranded DNA waiting to be unwound. The duplex conformation downstream is maintained by self-stabilizing base pairing as well as long-distance electrostatic and van der Waals interactions of the DNA with the RNA polymerase. Formation of the elongation complex does not involve major structural changes in the polymerase core. However, with the exit of the sigma factor, its contact sites, as has been already described in the previous video, now become available for interaction with the nucleic acids and establishment of a stable processive elongation complex. So, there are specific adjustments in the core enzyme conformation that meet requirements of stability and processivity. As for example, changes occur in a certain domain of the beta prime pincer, as shown by the red arrow. The details of the changes are shown in the next slide. Elements in this domain, such as an alpha helical coiled coil, a beta prime loop called the lid, and another loop called the rudder, all move closer to the DNA. The lid stacks with the upstream edge of the RNA-DNA hybrid, while the rudder gets into a position between the downstream DNA and the hybrid. Besides, a beta prime 3 helix bundle is involved in several polar and van der Waals interactions with the double-stranded DNA phosphates. A domain in the beta pincer that had earlier contacts with sigma now moves closer to the DNA-RNA hybrid. With all these movements, the RNA polymerase close in to reduce the dimension of the main channel. The surface compatibility between the protein and the nucleic acid is established, and thereby stability and processivity of the elongation complex are ensured. However, the major sources of stability of the elongation complex are polar and van der Waals interactions between the polymerase and the RNA-DNA hybrid backbone phosphates, supplemented by hydrogen bonds between the polymerase and single-stranded RNA in the exit channel. Processivity of the RNA polymerase enables it to undertake thousands of nucleotide addition cycles. But it must be remembered that the enzyme adds only one nucleotide to the nascent RNA in each cycle. For every nucleotide added, there is a forward translocation of the enzyme relative to the template by just one nucleotide. Separation of the downstream DNA by one base pair and displacement of one nucleotide of the nascent RNA from the DNA template at the upstream end of the RNA-DNA hybrid. Further, as the lids stack onto the upstream base pair of the hybrid, growth of the hybrid beyond 9 base pair is sterically hindered. A concerted motion of the bridge helix and the trigger loop, functioning as a BHTL unit, has an important role in the nucleotide addition cycle. When the NTP binding site is available for the next NTP, the polymerase is in open conformation, the trigger loop is unfolded, and the bridge helix is kinked. As the NTP binds, the trigger loop folds into a double helical structure. The bridge helix adopts a straighter conformation, and the polymerase goes to a closer conformation. As the incoming NTP is correctly positioned, when incorporation is complete, a pyrophosphate is released. The trigger loop unfolds, the bridge helix is kinked, and the polymerase readopts an open conformation. Now let us talk a little bit about transcription fidelity. The fidelity of the transcription process depends on three activities of the RNA polymerase complex: selection of the correct NTP substrate, detection of a misincorporation, and error removal by polymerase backtracking and RNA cleavage. Correctness of the NTP is primarily determined by Watson-Crick interaction with the template base. The trigger loop and a few surrounding residues assist in the selection. In case a non-complementary NTP is incorporated, one or a few RNA 3' end nucleotides are frayed away from the template and further closure of the active center is prevented. The polymerase is paused and there is no further extension of the nascent RNA. Now, for cleavage, the target phosphodiester bond must be aligned with the catalytic site. So, the RNA polymerase backtracks. The frayed and backtracked states display bent TL conformation. The misincorporated nucleotide is now prone to be removed by polymerase's intrinsic nucleolytic RNA cleavage activity. RNA cleavage is enhanced by stimulating factors. In E. coli, there are two such factors, GRE-A and GRE-B. GRE-A induced cleavage shortens RNA 3' end by 1 to 3 nucleotides, whereas GRE-B can cleave fragments longer than 3 nucleotides. The GRE factors reconfigure the active site into the cleavage mode. Once the error is removed, RNA synthesis resumes till the polymerase meets a terminator. Stability of the elongation complex is important for the RNA polymerase to undertake an efficient transcription process. So, transcription termination requires destabilization of the elongation complex. Now, there are two mechanisms that destabilize the elongation complex leading to termination. They are intrinsic and factor or row dependent. The canonical intrinsic terminator comprises two sequence elements: a short GC-rich inverted repeat of approximately 20 base pairs, immediately followed by a stretch of 7 to 8 80 base pairs. When these sequences are transcribed by the RNA polymerase, the RNA produced has the potential to form a stem loop structure or hairpin, which is followed by a 7 to 8 nucleotide U-rich tract. As the terminator U tract is synthesized, formation of a poorly stable RNA-DNA hybrid within the main channel leads to decreased elongation rate and subsequently RNA polymerase pausing. This provides time for the terminator hairpin to form at a stage when it can invade the RNA exit channel of the RNAP. The event triggers the elongation complex destabilization and finally dissociation. The second mechanism of transcription termination is executed by the homohexameric helicase Rho. The enzyme binds to substrate RNA. There are two levels of binding, primary and secondary. The primary binding is at rut sites of RNA. These rut sites contain pyrimidine dinucleotide interspersed by loops. Rho entity contains a narrow cleft which can accommodate a pyrimidine dinucleotide. The first pyrimidine is held within two tyrosine and a glutamate residues, while the second pyrimidine interacts with an arginine and an aspartate. The secondary binding sites in Rho CTD are the conserved Q and R loops. Now, compared to primary binding, the secondary binding is more non-specific. However, carbonyl group of Rho valine residue in Q loop hydrogen bonds with the 2' hydroxyl of RNA ribose. So, secondary binding is specific to RNA. The Rho CTD also contains Walker A and B motifs, including the P loop that create the ATP binding pocket. A glutamate residue and two arginine residues form the active ATP hydrolysis site. Now, driven by ATP hydrolysis, the enzyme translocates the nascent transcript. ATP hydrolysis further forces shearing of RNA-DNA hybrid, extraction of the nascent transcript, and collapse of the elongation complex, leading to the release of the DNA template. The materials contained in this presentation are taken mostly from the book Fundamentals of Molecular Structural Biology.